Okay, now this lecture focuses on the second part of the digestive system. Now, an introduction of this specific part is we're going to talk about the small intestine, the large intestine, and then we're also going to talk about the accessory organs like the liver, the pancreas, the gallbladder, and the rest of the accessory organs. Now, what we left off is last lecture we talked about the stomach. Now, the stomach is connected to the small intestine, so that's the next one we're going to be talking about. So the small intestine, what that means is just small bowel. Compared to the large intestine, this one is about smaller in diameter, but it is 6 meters long, so almost 20 feet, and you could say it is the largest portion of the alimentary canal. We might not feel like it is 6 meters long because, of course, it is crunched up and folded. Now, it has 4 major processes, which include secretion, digestion, absorption, and propulsion. Now, we do have these cells called enterocytes, which are the small intestine cells, that are going to produce multiple digestive enzymes, hormones, and also mucus. And we'll talk about them as we go along this second part. Now, these enzymes that are produced, they are also going to be released by the pancreas, which are going to be responsible for the bulking of chemical digestion. And we'll talk about more of that in detail. There's no further nutrient digestion that's going to take place by the human enzymes once the food leaves the small intestine. So this lets you know that most of the digestion happens in the small intestine. Now, bacteria that are found in the large intestine, they really generally don't digest it for certain nutrients compared to the ones that are in the small intestine. Now, after the nutrients are digested chemically, meaning with the process of enzymes, and they are absorbed across the enterocytes, which are the small intestine cells, then they're going to go through the blood, the lymph nodes, along with water, vitamins, and other substances that are also used for absorption. So the small intestine is mainly going to mix, it's going to propel, and it is going to pass its contents along the large intestine, which is connected to the small intestine. So the small intestine has 3 divisions, and they are distinguished based on their histological differences, so meaning their tissue differences. We learned that the duodenum is the first segment or the initial segment of the small intestine that is connected to the stomach. So here is the stomach, or here, and here we have the duodenum, which is the shortest segment of the small intestine of the 3, which is about 25 centimeters, 10 inches long, and if you notice, it forms a C shape. It curves around the pancreas. The pancreas will be kind of back here. I'm not sure if you could see it. It will be positioned here. And it is going to be considered a posterior to the perineal cavity. So it is considered retroperineal, meaning it is under the perineal cavity, which we learned from part number 1. Now it has houses major duodenal papilla, which are here. I'm not sure if you can see it. This is kind of like cutting and finding it inside. And this is where the secretions from the gallbladder and the pancreas enter the small intestine, which we'll talk more in detail once we talk about the gallbladder and the pancreas. The duodenum submucosa contains specialized glands, which are going to be called duodenal or Brenner's glands. And they're going to produce alkaline mucus, meaning a basic mucus, to protect the duodenum from the acidic chyme that is coming from the stomach. So it produces a basic substance to kind of help neutralize that acidic substance, which is the chyme that comes from the stomach. So that is the duodenum, the first part of the small intestine. Then the duodenum is connected to the jejunum, which is the green portion here. Here's more or less how it looks like mainly. It does have a duodenojejunal flexure, which is going to be in between the duodenum and then the jejunum as well. Now the jejunum in the green is going to be about 2.5 meters, 7.5 feet long, and it is where it's most active for chemical digestion and absorption. So here's where most of the enzymes are going to be released in, and the absorption is going to happen as well. The green portion here following the jejunum is going to be the ileum, which is going to be the final segment of the small intestine. This one is going to measure about 3.6 meters, which is about 10.8 feet in length, and it is going to finish in the cecum, which is the part that connects the small intestine to the large intestine. Now, this specific section here, the ileocecal valve, is going to prevent the secretions from the small intestine to go back to the previous parts, like go back to the ileum, and then flush up. This is very critical in preventing the access of the bacteria from the large intestine. So any bacteria that is in the large intestine cannot access the small intestine because of the ileocecal cave, which is, like I mentioned, the large portion of the small intestine, part of the ileum, right before it hits the large intestine. Now, the structure and functions of the small intestine, they are going to, like I said, mainly be for digestion and also for absorption. Now, this is what the small intestine looks like. And if you notice, it has these folds, okay, which we could see here. And if you notice, it's very thick. If you have dissected like an animal, like a frog or a pig, you're going to notice that it's a very thick lining. But it is going to have these circular folds, which are applique, and it is going to more or less look like this. Okay, you can't really see it. This is where you're going to find most of the enterocytes, which the enterocytes is where the absorption happens. The small intestine has two types of folds. They're not really visible with the naked eye, but these are going to be called villi. So the mucosa folds into projections called villi. So if you see it here, this is one villi, and then here is how they look like. They look like very thin mountains. And if you notice, it has a simpler columnar epithelial tissue, which if you notice it is thick, it is this part right here. It has goblet cells, which they look like the little circular ones. These are going to produce mucus. And then they have blood capillaries, which are here in the purple. We have the lymph nodes, and then we also have the arteries and the veins. Now, between the villi mucosa, there's indents and intestinal crypts, which are going to be these here, okay? And in these here, you have both the endocrine cells, which are the regular cells that look more like a cube, and then you also have the mucus goblet cells, which are the ones that are circular around. Okay. And so then aside from the intestinal crypts, I'm sorry, I kind of went ahead on in the previous slide, but the villi have something called microvilli. So if you take a chunk of the villi, you're going to notice that it has these endocyte, which is a small intestine cell, and it has looks like it has hairs. This is mainly the brush border, and one enterocyte, just as you know, it could have about 3,000 microvilli, which are very, very small here. And digestive enzymes are produced here. They are secreted by the endocytes themselves, and they're here to break down disaccharides and peptides. So the small intestine motility differs from fasting and eating because during fasting, the small intestine is slower since there's not really a lot of movement when the food is brought in. There's less rhythmic contractions, and they does it in a pattern called migrating motor complex. This is when the contractions are clear. They're clearing any of the remaining material since there's not anything coming in that is new. It is going to clear anything that is remaining, any leftover food or secretions from the previous meal. Now, this occurs about 2 hours before pushing the food from the duodenum to the ileocecal valve. And this process is called by the enteric nervous system and the hormone motilin, which is produced by the duodenal mucosa. Now, the motility of the small intestine, when I say motility, is a contraction in the movement of the food, undergoes 2 types of movements, and this is during eating when the food is passed down. The vagus nerve is going to appear to regulate both the peristalsis, which is the pinching of the muscle, and segmentation as well. So in terms of peristalsis, there is 2 types of um, alternated contractions, which are alongside the longitudinal and circular layers of the smooth muscle. The longitudinal one is this one here is kind of like the outermost, okay? And this, then you also have the circular, which is the inner one here, okay? They're mainly here, of course, for propelling from the chyme, which is a liquid food, towards the ileum, and then ultimately through the ileocecal valve, which connects the small intestine to the large intestine as well. So here in this image is showing how the longitudinal works. So longitudinal kind of pinches the overall, in this case, lining of the small intestine. And then the circular one is kind of pinching the cavity, and the circular chyming does, like the mixing of the chyme happens here. Now, segmentation, like I said, is internal churning, and it's only happening in the circular layer, like I was explaining here. And there's mixing, mainly this is mechanical digestion, that physical digestion, that in correlation with also intestinal and pancreatic enzymes as well. Now, hormones and pancreins, which are signaling pathways, that control the secretion and motility of the digestive organs, which is summarized here as well. Now, in terms of the hormones, how they space, what is their function? You have, of course, hormones that are only going to work in the stomach. You have gastrin, which is increases acid secretion by parietal cells. So we talked about in part 1. Histamine is going to increase also acid secretion by parietal cells, same thing as gastrin. Serotonin stimulates gastric motility, and somatostatin decreases the secretion by parietal cells, which mainly these we focus on in the part 1. Intestinal hormones, these are going to be the ones that we're going to mainly talk about in the second part. We have the cholecystokinin, which causes the gallbladder to contract, release bile, which we're going to learn what bile is exactly later on. Stimulates secretion of pancreatic enzymes from acinar cells and relaxes the hepatopancreatic sphincter. Like I said, we're going to learn about this as well. Gastric inhibitor peptide inhibits the secretion from parietal cells. Intestinal gastrin stimulates acid secretion from parietal cells as well. Motilin stimulates the migrating motor complex of the small intestine. Secretin is going to inhibit the gastric motility and acid secretion. It's also going to stimulate biocarbon ions that are going to also neutralize acidity. These are coming from the pancreatic duct cells and increases bile production by the liver. Lastly, we have vasoactive intestinal peptide, and these are going to inhibit the secretion by parietal cells. They also stimulate pancreatic secretion that increases the blood flow. Like I said, these are mainly going to be talked more in detail in this part. Okay, so now we're going to talk about the large intestine. The large intestine means large bowel, and it runs along the border of the abdominal cavity, surrounds the small intestine and other abdominal organs like the frame. So this is a large intestine, by the way. It has is bigger in diameter compared to the small intestine. It is about 1.5 meters long, about 5 feet long. And like I said, it has a larger diameter than the small intestine. The large intestine receives material from the small intestine. Anything that was not digested or absorbed it is going to then be considered feces or fecal matter, and it needs to exit the body. Now, in the large intestine, most of the water is going to be absorbed in the electrolytes. Now, these are going to be very critical fluids for maintaining electrolyte and acid-base homeostasis, meaning maintaining normal levels of pH in the body. Now, the task of the large intestine, just like the small intestine, is going to include secretion, mainly for mucus, propulsion, and defecation will be only unique for the large intestine. Now, it houses a lot of bacteria, numerous bacteria. When we think of bacteria, we think that it's bad, but in this case, these bacteria are going to perform important functions such as synthesizing vitamins that are crucial for cellular activities. Okay, so now we're going to talk about the different parts of the large intestine, its anatomical features, and it has different regions. We have the cecum, which is this part here that is going to bind the small intestine to the large intestine. It's here in the lower quadrant of the abdomen, and it is considered intraperitoneal, so meaning that it is going to be an organ that is completely surrounded by the peritoneum tissue, which the peritoneum tissue is a serous membrane that is found in the lining of the abdominal cavity, and like I said, it's covering some of the organs. So the cesium is going to be considered covering by the peritoneum. And then it features a smaller ended pouch, which is posterior. We're going to call it the vermiform appendix, or for short appendix, and it will be this part right here. This one, a lot of people say that it doesn't really have a function, but it does. It has lymphatic nodes that are going to help the immune system. So they harbor immune system qualities to help fight anything that pretty much you ate from the food. Some people have this removed and they still do pretty well as well. So then we have the cecum. Now we have the colon, and the colon is going to be the rest of the large intestine with the exception of the rectum. And the colon is divided into four portions. We have the ascending colon, and this one is retroperitoneal, meaning that it is going to be located behind the peritoneum, not surrounded by it, but behind it. And then you're going to have the hepatic flexure, which is here. It's kind of like the turn, so it turns to the left, you could say, like a left-hand turn. It is going to be now converting into the hepatic feature. Then the hepatic feature then moves to the transverse colon, which is here, which passes transversely across the superior abdominal cavity. Next, you have the transverse colon, then moves to the splenic flexure, the left upper part of the large intestine, and then you have the retroperitoneal descending colon, which is going to be found behind the peritoneum tissue. And then last but not least, you have the sigmoid colon, which is the bottom right, and is also intra peritoneal, meaning that it is found surrounding this part. The peritoneal tissue is surrounding this part. So the last part that I didn't mention, but you do have the rectum, which is the part that it connects to the sigmoid colon. And this is the large part of the large intestine. So the rectum is part of the large intestine. And it is going to be retroperitoneal, meaning that it is going to be behind the peritoneum tissue. Now, the rectum is going to have different valves. We're going to call them rectal valves. Okay. And these are valves that when there is some type of like flatus, like gas, that needs to go through, it goes through them. And this prevents any feces or fecal matter to go through it. So only the gas passes there as well. Now the rectum ends in the anal canal, which is here, and it is, you could say, a large portion of the intestine. I'm sorry, the last portion of the intestine. Now, the anal canal passes to the levator ani muscle, which is this one here. Okay. And the anal sphincter is another muscle as well. Sorry, the internal anal sphincter, which is going to be considered involuntary, meaning that you do not have any control of when this muscle contracts. It is controlled by the parasympathetic part of the nervous system, which is the one that is responsible for digestion of the body. You have the external anal sphincter, which is this one here. Okay, and this one is considered to be voluntary, meaning that you do have control of when this muscle contracts. For example, let's say that you have the urge to go to the bathroom, you kind of consciously, you voluntarily, you can pinch these muscles so that like you prevent feces from coming out. This will be part of the somatic motor neuron part of the nervous system. And then the large intestine also has the following histological features, different tissues. Unlike the small intestine, the large intestine doesn't have villi. It doesn't have microvilli. It does have some structural adaptations that reflect that type of function, meaning nutrient absorption, but it doesn't have those specific structures. It's enriched in mucus because it does have goblet cells, just like the small intestine, meaning they secrete protective mucus and lubricating as well. The muscular is external from the rectum. Is going to also have some type of longitudinal layer that is not continuous, but it's still most of the length. And it is gathered by three bands, or ribbons, you could call them the tini coli. And then some part of the small intestine does have these tension or bunches of the colon or pockets, which are going to be these little segments that you see here. Okay. They're going to, like I said, be causing constant tension and, help also with the movement as well. And then you have the serosa tissue, the visceral peritoneum, which is the connection between the peritoneum tissue and then an organ. The organ will be, of course, the large intestine, and is filled with fat-filled pouches, which we're going to call them epiploic appendages as well. But if you notice the keep saying small, large intestine does not have microvilli. It does have goblet cells. It does have mucus for lubricating as well, which, of course, mucus is more slippery for things to go through as well. Now, in terms of the bacteria, the large intestine does harbor a lot of bacteria. Just in fact, there's 10 more times more bacteria in the large intestine than the own cells, mainly because human cells are way larger than bacteria. So there's going to be 10 more times of bacteria in number. Just to give you an idea, bacteria make up as much as 60% of the dry mass of feces. So when you release feces, it's probably going to have most of the bacteria. Now, this bacteria is not going to be bad. It's part of the normal flora or the gut flora. It has about 500 different bacterial species, and they are coexisting with humans. So meaning bacteria and the human organism have a symbiotic relationship, mainly mutual, meaning that both benefit from the relationship. So humans are going to provide an environment for bacteria that is needed for them to survive, ideal temperature, pH, even they get the food from our food. And what bacteria are going to do is they're going to perform very useful functions, including producing vitamins. Very common one that some bacteria produces vitamin K, which is necessary for blood clotting and other mechanisms in the body. Aside from producing vitamins, bacteria also metabolize undigested materials. So there's some materials that our own enzymes from the digestive system cannot digest, but these bacteria, they can help with that. They can metabolize carbohydrates, which are soluble fibers that the small intestine cannot digest at all, and converting them into fatty acids, which are now easier to absorb by the small intestine. They also fight other bacteria that are harmful, so they fight them off. Any bacteria that are pathogenic or disease-causing. In the normal flora bacteria, they compete for nutrients, for space, and they even kill each other off. But only the harmful species. They even stimulate the immune system. So meaning that sometimes the bacteria, they activate the immune system, specifically, mainly during infancy. And they're going to also train the immune system in knowing is there anything foreign? Also know about antigens, which are found in bacteria and other foreign pathogenic organisms. And at the same time, they stimulate the development of mucosa-associated lymphoid tissue or MALT, which is going to then induce the production of antibodies against a pathogen, which is also part of an immune system mechanism. So bacteria are not all bad, okay? Now, same thing as the other organs, motility has to happen in order for the absorption of water and electrolytes to happen, of water as well, and then also defecation, anything that is not needed. So the large intestine has two functional segments. The proximal large intestine is going to consist of the ascending and transfer colon. So ascending is here, and the transfer colon is here. These are going to be mainly the distal part of the large intestine. And then, sorry, the proximal are going to be the ascending and the transverse, and the distal part of the large intestine are going to be the descending and the sigmoid colon, which is like on the right-hand side. The proximal large intestine, which is going to be the ascending and the transverse, that one is mainly for absorption of water and electrolytes, and also bacterial activity. Whatever the functions of the bacteria I just talked about, that's going to mainly happen here. So the proximal large intestine, ascending and transverse, they are going to exhibit motility in segmentation, meaning turning, the mixing of the chyme. And this is going to happen repeatedly. There's going to be swirls in there around the hastrum, which is these little segments right here, these little dividends there. Now, this also mechanism is going to aid water and electrolyte absorption. So this is where primarily the water and the electrolyte absorption happens. And it is controlled by neurons of the enteric nervous system, which are going to trigger a stretch, quote unquote. Other types of motility in the proximal large intestine, ascending and transfer colon, is going to be known as mass movement or mass peristalsis, where you have a massive movement, meaning multiple hastra undergo peristalsis, so each multiple ones of these segments. It is going to propel their contents into the distal large intestine, which is going to be the descending and the sigmoid colon part of the large intestine. These movements are going to be about 3 to 4 times per day. That's why, for example, when you're not eating or you're just sitting there, you feel like some type of movement in your large intestine, or sometimes it even makes sounds. This is what's happening. Now, these mouth movements, like I said, can trigger by food consumption when you're initiating the reflexes that are controlled by the enteric nervous system. So like I said, the distal large intestine is going to be the descending and the sigmoid colon. Now, this is going to perform a very small amount of absorption, mainly water, but the proximal part of the intestine does mean the main absorption. This part mainly is going to, main role is to store fecal material until it is ready to be expelled out during defecation. Okay, so most of the feces or fecal material are going to be on this part of the large intestine. And the distal large intestine is going to be less motile. It's not going to have those mass motility steps like the proximal one. But it is going to mainly be forcing fecal material into the rectum, where we know that it is controlled by the parasympathetic part of the brain, and this is called defecation reflex. Now, there is different steps that I'm about to explain, but there's stretchers, receptors that transmit the sensation of that there's fecal matter that needs to be removed. So distension is going to happen. So let me start explaining there as well. So the stretch receptors are going to then say, okay, there's distension, meaning they kind of are spread apart. So the linings here of the rectum, they're spread apart. This distension mechanism is going to then activate the receptors, send a signal to the spinal cord, okay? And then a signal is sent back by the parasympathetic system in saying that it needs to contract in order for the anal canal to do this type of reflux, okay? So they kind of separate apart. So then that is going to have another mechanism which is going to impulse. The brain already knows, okay, this tension's happening. We need to contract the extrinsic endosphincter and all the characteristics of the rectum to be able to release the fecal matter or the feces through the rectum. So this distension, like I said, is this process of where the uh, lining of the rectum kind of separates, and that allows more feces to go through. So it's like a contraction because it is muscles. These here represent muscles as well. Okay, so put everything together. Like I mentioned, the large intestine is the last part of the oral cavity. And anything that deals with motility of the large intestine, like I said, is going to, most of the water is going to be absorbed, but depending on how much water is absorbed, it can definitely affect the solidity, meaning if it's going to be more of a solid or a liquid in terms of the feces. So when motility increases, large intestine does not have enough time to absorb because it's kind of taking more time for motility instead of absorption. So you're going to notice that the feces is more watery, kind of like for diarrhea. And then factors that increase this motility will be irritation, like there's bacterial viral infections that this part of the body is trying to fight off, and it doesn't have time to absorb that much water. Or even drugs. And sometimes there is drugs like medication that also enhance this parasympathetic nervous system mechanism, and more water is going through the feces. When motility decreases, there's way more absorption. It absorbs so much water that the feces becomes harder and individuals become constipated because the feces is not soft as it needs to be. This lack of motility maybe slows down because of taking medication, maybe opiate narcotics, or some type of inducing mechanism that blocks the effects of the acetylcholine, which is a neurotransmitter that enhances this. So putting everything together, summarizing everything together, including the large intestine and how it's associated to the other organs in the alimentary canal. So once again, you have the oral cavity, which is mainly for ingestion mechanism, mechanical digestion like chewing, and then propulsion and secretion. Then it's going to go through the pharynx, the esophagus, and in the stomach, where propulsion, chemical digestion, mechanical digestion happens, secretion, and also limited absorption happens here. And then it's going to go through the small intestine, which is this portion here, smaller in diameter compared to the large intestine. Chemical, mechanical digestion happen. Most of the absorption happens here, secretion and propulsion. And then last but not least, you have the large intestine, where some absorption of the water, most of the absorption of the water happens, electrolytes, and then vitamins are produced here, propulsion, there is limited secretion, and then the last thing is defecation. So the alimentary canal is, all of these do come in contact with the food. Okay, so now we're going to move on to the accessory organs, okay? Most of the accessory organs do not come in contact with the food, with the exception of the teeth and the tongue, okay? Starting off with the salivary glands, okay, that produce mucus. And then we also have the pancreas, the liver, and the gallbladder, which we're going to talk more in detail in the next coming slides. Now, these are going to secrete exocrine glands. These are considered, I'm sorry, they have exocrine glands which produce those enzymes for chemical digestion. Like I said, they're happening, they're making these enzymes, but they don't touch, these organs do not touch the food. So they secrete products into the alimentary canal, and they're going to appear to be within the body, but the alimentary canal is only going to be open to the open environment, so kind of like the mouth and the anus. So the lining of the alimentary canal is considered to be external to the body surface. Okay, so first up we have the pancreas. It is an accessory organ in the pancreas. There is glands that produce endocrine hormones and exocrine, meaning the enzyme function. Now the endocrine functions are going to produce hormones like insulin and glucagon. Insulin is used to decrease sugar in the blood, and glucagon is to help increase sugar in the blood, like when you have low sugar. And they affect most of the cells in the body. So when these hormones are released, most of the cells are going to be affected by it. The exocrine secretions are going to be when enzymes are produced, and they're produced by clusters by acinar cells, which we're going to talk about what they are in just a moment. When they're released, they're releasing the ducts of the pancreas, and they're going to be emptied out into the alimentary canal. Now, the pancreas, it has a primary upper left quadrant of the abdomen and extends to the duodenum of this to the spleen. So the pancreas has three parts. It has the head, okay, which is the kind of like the thicker part, the body, which is the longer one, and then it even has a tail. Now the head is in contact with the duodenum. That's the initial part of the small intestine, okay. And then the body is going to be separated by the tail and it's towards the spleen, which the spleen is kind of over here. I'm sure you could see it. Now, the pancreatic duct, which is this one here, the main one, it looks like a vessel, but it's a duct. It's going to be, it goes along the length of the pancreas, and it is going to receive secretions from the acinar cells, which the acinar cells are going to be located here, okay, which are found throughout the whole pancreas. Okay, so talking more in detail, near the duodenum, you're going to have this main pancreatic duct. If you notice, it's here, and then it goes the whole length of the pancreas. And it is going to empty out into the duodenum, which is here. Now, the acinar cells, which are found here, they're going to be known as acini for plural, and they're going to be joining with the ducts, okay, which you hear, and anything that is secreted by them is going to go through and then reach the duodenum as well. So you could say that they're like ducts that are going to be secreting and dumping it in the duodenum. This one here is the gallbladder duct. Okay, here's a gallbladder in the green, and it also secretes and dumps secretions into the small intestine as well. Okay, so talking more about the bile duct, okay, there is something called pancreatic juice, kind of like stomach juice, but it is a liquid that is part of collective secretions of the pancreatic acinar and duct cells. Okay, so here are the cells, and they're going to consist of water and multiple digestive enzymes and other proteins. Okay, so the juice, the pancreatic juice, is dumped in here, and what produces that juice is going to be the pancreatic acinar and duct cells, which are found here. Okay, duct cells are going to produce bicarbonate ions, which bicarbonate is a base. And why is it produced like that is because it is trying to neutralize that acidic chyme that is coming from the stomach. So here's where the neutralization happens. So the duoduct cells produce that. And then you also have digestive enzymes that are produced by the acinar cells. And they're going to do the chemical digestion, mainly digesting carbohydrates, proteins, lipids, and nucleic acid as well. But they all are going to be produced here, and then they're going to go through the ducts and then be dumped into the small intestine. By the way, what produces the oh, it's in the next slide, I'm sorry. So then Q cells are going to produce these hormones that are going to enhance the pancreatic juice. So you have the secretin, which is produced by the duodenal cells. These are the duodenal cells, which are here. Some of them are going to release secretin, which is released by the duodenal cells, and it is going to be in response to acid and lipids of the duodenum, and they're going to trigger the duct cells to secrete biocarbon ions. And then you have the duodenal cells, which also release the chlorocytokinin CCK hormone that is also going to be inducing the duodenum, produced by the duodenal endocrine cells. And in response to the presence of lipids, they partially digest proteins in the duodenum as well. And they're going to activate the acinar cells that are going to trigger secretion of enzymes and other proteins. So these duodenal cells release secretin and CCK. They're going to go through the blood vessel, activate the endocrine cells and the duodenal cells as well. And then they're going to either produce enzymes or release biocarbon ions, which are going to then neutralize that acidity. Okay, next we're going to talk about the liver and the gallbladder. Now, the liver is going to have a right quadrant, okay, or we're going to call it a right lobe, and then it has a left lobe. The right lobe, of course, is thicker than the left lobe, and they're going to be connected by falciparum ligament, okay, which is a ligament like that ties organs to the bone, and is also going to be anchored by an anterior abdominal wall. You also have the round ligament here, and then if you notice, the gallbladder is right underneath. So when you flip the liver, now we have the right lobe here and the left one here. So let's keep it as like looking at the back part of the liver. It is going to have different veins, different vessels that are going to go through it. So you have, before I go over the vessels, aside from the right lobe, you also have the quieted lobe, which is on kind of like attached to the right lobe. And then you have the quadrant lobe, which is kind of next in between the round ligament and the gallbladder. Okay. Now in between here, you do have the hepatic vein, the inferior vena cava, which is here. The gallbladder on its own is going to be connected to where the bile duct will be released. We also have the porta hepatis, which has the hepatic portal vein. And then it has the hepatic artery, which is in the red. And then it has the hepatic, common hepatic duct, which is in the green. So if you notice, you have the gallbladder's duct that are connected, but also the veins and the arteries. And they're going to be used to release those enzymes into the blood system. Okay, so the liver has something called a liver lobule, which is a basic unit of the liver, and it has specific histological features. So let's say here that we take a part of the liver, you know, I'm sure we could see it here, okay, and we were able to zoom in. This is what it looks like. Okay, it looks like it is studded with a lot of vessels. So these histological features are going to be separated by one another by the septum, which is a connective tissue capsule of the liver, and it is composed of flattened plates or cells that look like flattened plates, which we're going to call hepatocytes. So each of these little segments that you see here represent a cell, which is hepatocytes. If you zoom in, each one is going to be that hepatocyte, a cell, and the purple here represents a nucleus. They have like a hexagon shape. I'm sorry, they're staggered in a hexagon shape, like you see here. Kind of, you could see that it's a hexagon. And there's levels. So here we have different levels that are stacked upon themselves, and then you have a thick layer of those. So then this arrangement has something called the portal triad. And triad because it has three vessels that are part of this component. You have the hepatic arteriole, which is in the red. You have the portal venule, which is in the purple, and then you have the bile duct here. So the vein, the artery, the venule, and then the duct, if you notice, are connected as well. The portal venule and the hepatic arteriole, they're kind of connected into a capillary. But that's why they call triad, because there's three vessels or linings there as well. And then the hepatic arterioles and portal venule, they both drain into the large and leaky capillaries. Like here we see them better. Called the hepatic sinusoids, which they pass between the rows of the hepatocytes. So they kind of they will be here. Okay. Now, why is these specific liver lobules studied with these vessels? Is because, of course, blood flows through there, and materials are exchanged between the blood and the hepatocytes, and they're eventually draining into the central vein, which then travels through the heart, and then the whole blood flow happens that way. So there's a central vein here, okay, and this vein is going to then fit into the inner vena cava, which is then it's going to go to the heart. Now the bile, how it flows through the liver of the lobule to the opposite direction, is going to be also from the hepatocytes into the tiny ducts, which we're going to call bicaniculae. And they're represented here. I'm not sure if you could see them, but they're here, and they're found throughout the lobule. And these are eventually going to drain into the bile duct. Okay, so talking more in detail about the function of the liver, like I said, it is an organ. It doesn't really come in contact with the food. That's why it's called an accessory organ, but it is no more important. So the liver has one or more diverse sets of functions, like any other organ, but it's going to release both endocrine, meaning hormones, and exocrine secretions, meaning the enzymes, to convert harmful chemicals into non-toxic substances that the body can eliminate. So think of your liver as neutralizing any toxin that you introduce into your digestive system. It is going to be neutralized there. Now, the main digestive function of the is to produce bile, because bile contains water, electrolytes, and other organic compounds. Bile is going to serve two critical functions that are required for the digestion and absorptions of lipids, meaning fats. And the mechanism by which the liver excretes the waste in other substances is going to be done by the liver instead of the kidneys, which they can't do. So bile salts are going to be one of the bile's main organic compounds that is produced by it. It is derived from cholesterol, and bile salts have a feature called amphiphilic, meaning they have both polar and nonpolar parts, meaning hydrophilic, hydrophobic. Some parts are going to interact with the lipids, meaning the nonpolar parts, and the other ones are going to interact with the watery environment of the small intestine, meaning that polar part. So these are coats of lipids that are found in the duodenum. They're going to be physically broken apart into smaller pieces. This mechanism is called emulsification. This is some type of mechanical digestion because something physically is broken. In order for chemical digestion, meaning the enzymes to start the digestion and absorption of the lipids happens that way. Bile is also contains, by the way, bile is a secretion, contains a lot of materials that the liver excretes, including cholesterol, waste products, and even toxins like heavy metals that are normally neutralized by the liver. Most of these products are not reabsorbed by the large or small intestine. They're supposed to mainly be part of the feces, meaning expelled out of the body. So how the liver is going to break down or secrete that specific mechanism is going to mainly be hepatocytes. They secrete bilirubin, which is going to be a hormone that is going to break down hemoglobin, which hemoglobin is found in the protein found in the red blood cells. This is going to be done by the spleen. Okay, so hepatocytes secrete bilirubin, which is put into the bile. Then the normal flora in the large intestine is going to convert it into urobilinogen, then into stercobilin, which then that is responsible for giving it the brown color of the feces. Okay, so if you have that brown color of the feces, that means that you have a normal color. More or less, digestion is happening great. If the feces is like black, that means it's probably there's like bleeding somewhere. But normally it should be more or less like a brown color. Some of the rubilogen is reabsorbed by the large intestine and then ends back by the hepatocyte portal system, and then it gets reused and resecreted into the bile. Some of the remains are in the blood and they excreted by the kidneys. Now, this is mainly going to give that yellow color of the urine, okay, that remaining going through the kidneys. The liver performs other functions, including nutrient metabolism, so a lot of nutrients are processed here. Detoxification, meaning releasing or detoxifying the body and releasing anything that is going to be harmful. Anything that we eat or drink is going to detoxify it from the body. Then, of course, excretion, anything that needs to be directly excreted. Bilirubin in the bile is going to, with other substances from the liver, are going to also enhance that. Specifically, antibodies. Antibodies are going to be proteins that are going to fight pathogens, and these are going to also be excreted by the specific mechanism. Also the kidneys. Okay, so now we get to the gallbladder. And the gallbladder is going to be this green, more or less it's green, small sac. If you touch it, it's very flexible. It looks like a balloon. It is going to be right under the liver. You're going to have to pick up the liver to be able to see it, and it will be here. Now, the function of the gallbladder is to store bile that is produced by the liver, and it is going to concentrate it, meaning removing any water, and releasing it when a stimulated CCK, the chlorocytokinin hormone is released. It's going to trigger contraction of the smooth muscle of the lining of the gallbladder. And then the gallbladder is going to then release it into a bile called cystic duct. So it will be this here. This duct is then going to be connected to the duodenum, which is where that bile is released. Now the cystic duct is going to also join the hepatic duct from the common bile duct, and then the remain is going to then join the pancreatic duct, which is here, and then they're going to release whatever they need. Now there's something called the ampulla, which is going to surround the ring of the smooth muscle of the hepatopancreatic sphincter, which is this one here. And then both of them are going to be dumped into the duodenum. Okay. Now, when bile contains calcium salts because of diet and cholesterol, they're going to be both then precipitate and form into heart lumps, which is called gallstones. If you're familiar with that, they are solidified components that are very hard to go through, and sometimes when you're trying to urinate, it hurts. Here's an x-ray of an individual, and here's what the gallbladder looks like. And if you notice, it has these little dots or circular structures. Those are the gallstones, which are solidified calcium salts and cholesterol. Now the liver produces bile, and it is going to be stored in the gallbladder. Now, when the liver produces bile continuously, it is generally not going to secrete it at a basal rate. Instead, the secretion occurs in responses to the gallbladder contraction. So depending on how the gallbladder contracts and their sphincter relaxation, then and at the hepatopancreatic ampulla, then it's going to be secreted, but at a basal. It's not going to be doing it at a basal rate. Now, this activity is mainly accomplished by the cholecystokinin hormone, so it is going to be controlled by that. And then, of course, the vagus nerve also plays a role in that. Other factors that influence bile secretion include the hormone secretin, which stimulates the production and release of hepatocytes, which are part of the lobus of the liver. So how does that work is, once again, you have your duodenal cells, which are part of the small intestine. They're here. And then they're going to, secretin and CCK, they're going to be released by the duodenal cells. They're going to go through the blood system. Then the CCK is going to trigger the contraction of the gallbladder. So it's going to be contracting, which is going to start releasing the bile into the duodenum. So then that's going to happen, and then the bile is going to be released in. Okay. Once that happens, it's going to be something called positive feedback, where the hepatoportal venule releases more bile salts, and then that means that the positive feedback meaning more contraction and more releasing of bile into the small intestine. So this is going to happen. It's going to be continuous motion, bile secretion, until the duodenum empties, meaning there's nothing more in the duodenum. At this point, the CCK, meaning the hormone, starts declining, and then the whole process stops. But this has to happen after the duodenum is emptied out. Okay, so this image here is going to now summarize the accessory organs, okay, part of the digestive system. So you have the teeth, which is where mechanical, physical digestion happens, mastication, chewing. You also have the tongue, where also mechanical digestion happens, propulsion, swallowing. The tongue does help with that. And then, of course, they have the taste buds, the sense of taste, those receptors on the tongue. You have the salivary glands, which you can't really see compared to the teeth and tongue, but they are there. They're going to secrete saliva. They're also going to assist with chemical digestion, meaning enzymes will be released by when the saliva. You're also going to break down or try to deter harmful microorganisms. It helps moisturize the food so it can be swallowed and go through the alimentary canal. And there also chemical digestion of the carbohydrates is going to start here as well. We also have the pancreas, which is also an accessory organ. It secretes and catalyzes chemical digestion of lipids and carbohydrates, proteins, and nucleic acids. It also secretes biocarbon ions, which neutralize the acidity of the chyme that was coming from the stomach. You have the liver, which we just learned that mechanical digestion happens. There's some enzymes that are released, specifically with bile production. It is going to excrete the waste from the bile. Anything that is wasteful from the bile is going to excrete it out. And then you have the gallbladder, which releases enzymes for mechanical digestion. It's going to store, concentrate, and release bile that was produced from the liver. Now, the process of digestion and breaking down the food into smaller pieces is going to be done until only individual nutrient molecules remain. So the smallest individual molecule is the main goal, which is what's going to be absorbed by the blood or the blood vessels. Now there's two types of digestion. Like I said, you have mechanical digestion, where there's physically something is broken into smaller parts. And then you have mechanical digestion, where enzymes break the chemical bonds of the food and it's even broken further. All nutrients go through the same mechanism of mechanical digestion, meaning mastication in the mouth, churning in the stomach, and segmentation in the small intestine. Chemical digestion is going to be a little different depending on the organs, what enzymes they produce, and what is needed to be absorbed as well, depending on that. But most digestive enzymes, they're going to be catalyzed by hydrolysis reaction. What is a hydrolysis reaction? Is when you need a water molecule to break something into two smaller molecules. So how that happens is that here we have a dipeptide, two amino acids. We need to break them down because digestion is happening. You have a small little water molecule that breaks the bond here. Now you have two separate amino acids in this case, alanine and glycine. But if you notice, the water components took place of where the two, in this case, amino acids were joined together. H2O, meaning 2 hydrogen, 1 oxygen. OH is going to go through over here, and one of the hydrogens is going to go here. Okay. Now, that's why so much water is secreted with fluids. That's why you need so much water in digestion, because you need a water molecule to break big molecules into smaller ones, like this example here. Now, digestive enzymes are going to speed up, of course, the reactions of the breaking down of these molecules. This is going to be called enzymatic hydrolysis, where enzymes use water to be able to break down these molecules into smaller ones as well. Okay, so now we're going to talk about understanding how absorption happens in the alimentary canal, specifically how does the food get into the blood system? How does the food get broken down into smaller molecules, and how does it get the blood system? Where there's once the nutrients are digested into the smaller components, now they're going to enter the body, meaning the blood system. Mucosa of the alimentary canal is going to be considered an external membrane. And ingested food is technically not into the body yet until it has been absorbed by the blood vessels. Okay. Nutrients and other molecules must be absorbed across the epithelial lining of the alimentary canal. After that, they enter the blood system, and then they can be delivered to whatever cell of the body needs those nutrients. Okay. So, for example, here we have the lining of the small intestine. This is the inside of the small intestine. You have the epithelial lining of the alimentary canal, and then you have the blood vessels. Okay. Now, when we're looking at the position of the enterocytes, the cells that are part of the lining of the alimentary canal, this is what the enterocyte looks like. The apical side, meaning facing the organ or the inside of the organ, in this case the small intestine, is going to be the apical side. So the apical side of the plasma membrane of the enterocyte is going to be facing the organ, in this case the small intestine. And then the basal side is going to be facing towards the blood vessel or away from the middle of the organ, which in this case is the small intestine. So how does it happen? Once again, in terms of proteins, I'm sorry, carbohydrates. So in terms of carbohydrates, how are they going to be absorbed into the blood capillaries? Okay, so in such proteins, if there is no proteins that are going to help the transportation, then they have to go through the lumen by themselves. But sometimes there is proteins that serve as transporters to help bring in the carbohydrate into the endocyte, and then that way it can go through the blood vessel. Okay. Now, sometimes when the enterocyte doesn't have those transporters, then the component cannot cross it. For example, mercury is a liquid mercury specifically. It could be very harmful. There is no transporter or no protein that can help transport it in, so it's going to stay out. Okay. So that's good for the body because if it's not supposed to go in, it's not going to go through because it can't cross it on its own. It needs a transporter like a protein to help it transport. But the endocytes lack that mechanism, so it's going to not allow them. If you have a transporter, then it could go inside of the enterocyte, okay, and then to the blood system. Okay, so the digestion of the polysaccharides happens in the mouth because in the mouth the saliva has this enzyme called salivary amylase, and the salivary amylase is going to have these digestive enzymes that are going to start breaking down the carbohydrate, meaning breaking down the polysaccharides, meaning many saccharides, many sugars, into few sugar oligosaccharides. Okay, so here we have a polysaccharide. Each one represents a monosaccharide, meaning a sugar, and then because of it needs to be broken down even further into an oligocyte. That was done by the salivary amylase. We need to break it down into individual sugars like glucose, galactose, and fructose. And that is how the sugars, in this case carbohydrates are made up of a bunch of sugars like glucose, galactose, and fructose. That is how they go through the enterocyte and into the blood system. So salivary amylase, like I said, is going to start in the mouth, continue in the stomach, And then since it's so acidic in the stomach, it's going to be inactivated. So they have to have further carbohydrate digestion in order for it to happen. So polysaccharides are broken down into oligosaccharides by pancreatic amylase. Okay, so let's say you have an oligosaccharide. The oligosaccharide needs to be broken down even further in order for it to go through the lining of the small intestine. That can include lactase, maltase, and sucrase. These are all oligosaccharides. Lactase is going to catalyze only the digestion of sugar lactose, like milk and milk-based products. And lactose is then going to be broken down into glucose and galactose. Okay, both maltase and sucrase are then going to be broken down into oligosaccharides, disaccharides, and then monosaccharides. And these monosaccharides like glucose, fructose, and galactose are the only ones that can go through the enterocyte and then through the blood system. So the carbohydrates start off as being a polysaccharide, then an oligosaccharide, and then broken down into galactose, glucose, and fructose. Then that is a small subunit of carbohydrates. That is how sugars are absorbed. So after carbohydrates are digested into the monosaccharides, and they're going to be absorbed by the enterocyte, go through the blood system that way. Okay. So in terms of glucose and galactose, they are the smaller subunits of carbohydrates. They are going to go against their concentration gradient, meaning they're going from a low concentration to a high concentration. When that happens, you need energy in order for this whole transportation to happen or absorption to happen. So there is something called a sodium glucose transporter, which looks like this in the blue. It is a protein, and it is going to be activated by a sodium potassium pump. The sodium potassium pump is another protein that also requires energy, which brings in potassium in the enterocyte, brings sodium out of the enterocyte. This is changing the concentration gradient of the enterocyte, so meaning it's the electricity of the enterocyte is changed. Once that happens, now the sodium glucose transporter can bring in glucose and galactose into the enterocyte, and then now it can be absorbed in the blood. So this whole process is a little more complex because you need the sodium glucose transporter plus the potassium and sodium pump, which this one drives the sodium glucose transporter, and then glucose and galactose can go through the enterocyte and then be absorbed by the blood system. Now, in terms of fructose doesn't really need the sodium glucose transporter. Instead, it uses another protein, but this process is called facilitated diffusion or passive process, meaning that it does not require energy. It is not going against its concentration gradient. It does not need that potassium-sodium pump. Instead, it goes through very nicely with the help of that protein, and then it just goes through the blood system. Okay, all three of these monosaccharides, glucose, galactose, and fructose, they're going to start in the apical side, meaning the enterocyte is facing the small intestine, and then leave the blood vessel in through the basal side, which is the one that is facing the blood vessel. Now, in terms of digestion of the proteins, now this one is also a little complicated because the enzyme that digests the proteins is called pepsin, but pepsin is not activated until there's a low pH. There's these cells called chief cells, part of the gastric glands, that produce the inactive form of pepsin, which is going to be called pepsinogen. Pepsinogen is made and is the inactive form of pepsin, meaning that when it is made, it is not going to start breaking any proteins. So pepsinogen, once it is in the pH of 2, which is very acidic, is now going to be activated and become pepsin. And when pepsin is inactivated is when the pH raises out to a level of 7. Now, why is pepsin made in inactivated form? Is because if it is active, it is going to start digesting the proteins, no matter where, to the point where it can even start affecting the organs, like the small intestine and everything. Only when it is in the pH of 2, which is very acidic, it starts activating it. Pepsin reactions, they're catalyze only about 10 to 50% of proteins of the ingested food. And the rest, the majority, is going to happen in the small intestine. That is going to help the pancreatic enzymes and the brush border enzymes. Now, these 5 pancreatic enzymes are going to digest the proteins, but they're going to be released in a not activated, meaning in inactivated form. Like I said, the precursor or the that is going to activate it is trypsinogen, that is going to now become into trypsin, and that's also going to be the activated form of the enzyme. Like I said, these enzymes are catalyze the digestion of proteins and polypeptides, which polypeptides are part of proteins, into oligopeptides, meaning few chunks of proteins, and then finally into free amino acids, which is the smallest subunit of proteins. Proteins are made up of amino acids. So how does the digestion of the amino acids happen? Like I said, you have the pepsinogen protein that is inactivated. Once it is in acidic pH, it converts into pepsin, and then that is how the digestion happens. So here you have the polypeptide or the peptide, and we notice it has a bunch of amino acids. Okay. You then release the enzyme to be able to break them down into free amino acids. There's also a protein that brings in sodium in the enteroside. At the same time, the free amino acid is brought in. You do need the help of the potassium-sodium pump to be able to activate this mechanism. And then the free amino acid then goes through another transporter in the green, and then it's going to go through the vessel. So this one needs more proteins. You have the transporter, the sodium-potassium pump, and this other one here that is in the basal part of the enteroside that is going to then be used as a transporter to go in the blood as well. Now, to be absorbed in the small intestine, like I said, proteins have to be broken down into free amino acids as a smaller subunit. And they also go into the Peyer's patches, which is part of the small intestine, and they're going to be part of the lymphatic tissue, which is trying to fight any harmful bacteria, any foreign pathogen as well. So I kind of already mentioned it already, but for the free amino acids to be absorbed, you do need the sodium-potassium pump that is going to then help release the free amino acids into the enteroside. They're going to exit into facilitated diffusion, which is another protein that is going to be part of the basal side, and then it's going to be released into the blood vessel. Like carbohydrates, amino acids are then delivered to the liver, and then they're going to go through processing via hepatic portal vein. Okay, so in terms of lipids, majority, actually 90% of the lipids that are taken down by your diet, they're going to be triglycerides, meaning meaning it's 3 fatty acids, which is a carboxyl hydrocarbon structure, that are bind to a glycerol, and they need to be broken down into smaller subunits. Okay, so lipids are nonpolar, meaning they're insoluble in water, but they do have a type of hydrophilic portion, meaning amphiphilic structures, meaning they have polar and nonpolar parts. So, for example, here we have what the bile cell formula looks like. You have the nonpolar part, okay, and then you have the polar part, which anything that is polar is some type of like fully charged or partially charged. In this case, we have the sodium that is charged here. So that is going to be polar, interacting with water, and the non-polar, they don't have a charge, they're neutral, then they're not going to interact with water. Okay, so to put everything together in terms of the bile salt mix, remember bile is made in the liver, stored in the gallbladder, and the bile salts, in the case of lipids, they're going to do some type of emulsification, meaning breaking them down into smaller pieces. And the end result is to get the smallest subunit of lipids, so it could be then emulsified, and then it could go through the absorption process. Now, this process of the lipid digestion proceeds in breaking down the lipids by the stomach churning and broken down by gastric lipids, that enzyme. Then the lipids are going to enter the small intestine. They're going to be emulsified by bile salts. Then you have another enzyme, pancreatic lipase, catalyzes the digestion of the lipids even further into free fatty acids and monoglycerides. So bile salts are going to remain associated with the digestive lipids and form micelles. So let me explain that in this image here. So here you have the stomach, that you have the churning is happening. You have the lipid globule, it's very large. So you have the gastric lipase in the purple. That if you notice, it broke that big globule. And then they're going to partially digest it into free fatty acids and glycerols. These are still partially digested. The glycerols have to be digested even further. So then once they are partially digested, then they're going to go through the small intestine. Okay. In the small intestine, you have the pancreatic lipase. Okay. Which before that you have the bile salts. The bile salts are then going to be considered emulsified lipids. Then you have the pancreatic lipases, which break it down into free fatty acids into monoglycerides. And now you have free fatty acids, monoglycerides. So then you have the bile cells that even digest the monoglycerides even further into the micelles. So then how the micelles get absorbed is here you have the micelle. This is the enterocyte. This is the inside of the small intestine. Now the micelles are going to then be broken apart. The free fatty acids or free lipids, are going to go in the enterocyte. Inside the enterocyte, the lipid is going to be surrounded by this chylomicron structure, or chylomicrons. And if you notice now it is surrounded, then it is going to be leaving the enterocyte into the thoracic duct, which is then going to join the blood in the rest of the lymph and thoracic ducts. And then the lipids are going to enter the cells where they are going to be used for different purposes. So you can see that lipids are a little complex because they need to be broken down further. The micelles are then going to be broken down even further for the lipid to enter the enterocyte. But the lipids don't travel on their own. They are going to be converted into chylomicrons, which that is how they travel the ducts. Okay. So just to summarize, the digestion and absorption of carbohydrates, proteins, and lipids. So carbohydrates are polysaccharides that are made up of sugars or monosaccharides. So the polysaccharide gets broken down into oligosaccharides by salivary amylase starting in the mouth. And then the polysaccharides are then even digested further into smaller oligosaccharides. And then lastly into monosaccharide, and that is what is absorbed by the blood vessels. Proteins need to be digested into polypeptides by pancreatic enzymes. Then they're going to be put into oligopeptides, and then free amino acids, those are the ones that are absorbed. Lipids, they are going to be digested by gastric lipase. They're going to be put into smaller lipid globules. You're going to have diglycerides, which are two fatty acids attached to a glycerol. They're going to be then put into micelles. Then you have pancreatic lipase, which is going to then digest them into free fatty acids and monoglycerides, which that is what travels in the blood. Now, just to give you an idea for nucleic acids, there is also chemical digestion that happens. Nucleic acids are like DNA and RNA. The enzymes that digest nucleic acids are nucleases. And they're going to break down nucleic acids into individual nucleotides, meaning adenine, guanine, cytosine, adenine, thymine, uracil, cytosine, and guanine, A, T, C, G, and U. Now, part of these nucleotides, they have a phosphate group, they have a ribose in case of RNA, or deoxyribose, which are both sugars, and then you have the nitrogenous base. So these are going to be the smaller subunits of how the nucleotides are digested, and then that is what is absorbed in the capillaries in the villi of the small intestine. So nucleases, they are made in the pancreatic juice, and they are going to digest nucleic acids. Lipids, the enzymes that digest them, are gastric lipases, pancreatic lipases. Carbohydrates, you have salivary amylase, pancreatic amylase, and then you have maltase, sucrase, and lactase, which are going to break down maltose, sucrose, and lactose into monosaccharides. And then the enzymes that digest proteins are pepsin, trypsin, chymotrypsin, carboxypeptidase, and then dipepsis and tripeptidase as well. Okay, so just to tie everything together in terms of absorption, in reference to water, electrolytes, and vitamins. So the alimentary canal absorbs a large quantity of water, electrolytes, and vitamins. And yet much of the absorption happens in the small intestine, but only a specific part of water absorption happens there, meaning water is going to be absorbed in the large intestine. Just to give you an idea, on average, 9 liters of water that enter the small intestine each day. 2 of them are going to be ingested, but the remaining 7 liters are going to be secreted into the alimentary canal and into the accessory organs as well. So out of these 9 liters, 8 liters are absorbed into the enterocytes of small intestine, because remember the water kind of breaks the molecules into smaller subunits. Most of the water absorbed in the enterocytes of the large intestine is gonna leave, but only about 0.1 of the water is being excreted in the feces. So not 0.1%, 0.1 liter is a very small percentage that when you drink water, it gets released in the feces. So water absorption is exclusively happening by osmosis, the traveling of water in and out of the cells, and it requires a concentration gradient, meaning that the enterocyte, which is a cell, is going to work with the extracellular fluid of the villi, and then it is going to drive the water movement. Anything that causes excess of solids remain in the lumen, and then the intestines are going to hold the water to prevent reabsorption that way. So osmosis is a process of water in and out of the cells, which cells are going to be the enterocytes. So electrolytes. Electrolytes are mini ions that are going to be taken from the diet, whatever you eat, and they're going to be present in secretions of digestive organs. Most of the electrolytes are absorbed in the small intestine, although significant amounts are absorbed in the large intestine as well. There's important mechanisms for absorption of these sodium ions that revolves in the co-transport. So they do have some type of co-transports with monosaccharides, which are part of carbohydrates, and amino acids, which are part of proteins. The sodium ion absorption is also a key role in anions, which are also charged. As sodium ions enter enterocytes, there has to be an electrogradient that is created for the driven of absorption of these ions, like chloride and bicarbonate ions. Other electrolytes are specialized mechanisms for their absorption. They include certain electrolytes, and they are present in smaller amounts, such as iron, magnesium ions that are absorbed by other mechanisms of transportation. Vitamins are going to be chemically generated. Mainly you get them from your diet. Some of the bacteria produce them as well. But vitamins, they have very important reactions in the body, which we're going to not talk about in this chapter, but there's 2 types of vitamins: water-soluble vitamins, meaning that they could be around water, they're polar, or fat-soluble vitamins that they need to be transported by a lipid because they are non-polar, they don't interact with water. Most soluble vitamins are absorbed in the small intestine by diffusion through enterocytes, through the enterocytes of the plasma membranes. Just to give you an example, one important exception is vitamin B12. This one does have to bind to intrinsic factors that are going to be produced by parietal cells of the stomach. So they are first in the stomach, then they are absorbed in the ileum, which is part of the small intestine. Fat-soluble vitamins, they are going to be packaged into micelles, which are lipid, specifically fats and other lipids that are going to be absorbed that way. So they need those lipid transporting structures to deliver them. Those include vitamins A, D, E, and K. And then coming to the last slide, just to summarize everything, the digestive system is a very important system because that is how your body gets all the nutrients, everything that it is needed for all the cells of the body to do their mechanisms. It starts off by the alimentary canal. You have the teeth, the tongue, the salivary glands that are going to start that mechanical and chemical digestion. Then the food travels through the pharynx, the esophagus, and then it's going to reach the stomach. In the stomach, you have also chemical and mechanical digestion. Here's where the food is turned into chyme, which is a liquid portion. And then it is going to go through the small intestine, the duodenum, where the gallbladder and the pancreas are going to secrete those enzymes for the even further digestion. Then they're going to go through the rest of the small intestine, where the breaking down of the proteins, carbohydrates, and lipids is going to happen even further. Absorption, the smaller subunits of the food are then going to travel the enterocytes, go through the blood vessels, and then be delivered to the ideal cells. And then anything that is left is going to go through the large intestine. Water, electrolytes are going to be absorbed. And then if anything needs to be released out, any waste is going to go through the rectum, and then finally the anal canal, and then expelled out of the body.